Am I supposed to do like the camera guy?
No. Action.
Alright, so where's the camera? This one, right?
Yeah, the one that says.
Okay.
Oh, I get it now. It's all your don't change anything.
Alright, you guys, so I brought some food for you guys. You
Yeah.
guys entertain. Yeah, so I have chicken wings and grapes. So
Aww.
here you guys go.
Have you tried the cotton candy grapes?
I have. They're crazy.
They are.
They actually taste like cotton candy.
No way.
No, they're crazy. They're green. Yeah, they actually taste like cotton candy.
Thank you.
What do we?
Alright, so can someone help me?
Yeah, what's up?
It's fixed. If you can, yeah.
Alright.
It's just a YouTube.
It should be one of the
Yeah, I got it.
fries.
Yes, yes.
Pass them
Never
around.
say no to fries.
No.
It's an evil smile.
Fries complete a meal.
Hey Nia, I never asked you like how to write your Chinese last name though.
It's just sweet ganza.
Without the top parts.
No, sweet. No, it's not tian. It's gan.
I know.
Right. So like,
And then the first name. How does you write it?
it's ning. So
Oh, it's Ning. Then why don't you spell like an I?
Oh yeah, it's spelled
NG?
wrong. It's because my parents didn't know English, and so they wrote on the birth certificate.
Oh, I got you.
Oh.
That's right.
Yeah.
I was like, always try to figure
Exactly.
out how
So
to write
there's a
it.
G at the end, right? It's
Okay.
ing, but it's
That's what I
Ning
thought.
Yeah.
So whatever. I mean, it's cooler anyways because nin is palindromic, and ning is
Yeah.
Yeah, so I like that mistake.
That's cool.
Let's get 6 letters. Okay, so anyways,
So now you have no secret. We know everything about you now.
no, not everything, because you don't, dude, I think I know more about PAHs than you know.
Ooh, wow.
Yeah. No, I'm
Sounds
serious.
sounds like a challenge.
Yeah.
Mighty words.
No, I'm serious, just listen, okay.
Bring it up, man.
Okay, so anyways, I'm Nin, obviously, right? I'm back again with another paper. I haven't done this in a long time. But this time we're going to be talking about oysters being used as a bioindicator for environmental PAHs. Okay, so
What's the full name for PAHs?
I'll get into that.
Ah.
I got this. Okay, so to do that, before we do that, I'm just going to go over some key words. The first one is biomonitoring. It's basically the act of looking at an environmental change or anthropogenic effect or some sort of pollutant in the ecosystem by using the biota. Okay, so you want to use the organism or a physiological process to do that. Okay, and to do so, you need to have a bioindicator. Right, an indicator species to hopefully respond to that environmental change in some way that you can figure it out. Okay, so I put a polychaete here, a sabellid, because this is actually from my master's research. I looked at how sabellids react to wastewater treated wastewater into the.
Yeah, I think recently there's gonna be a talk with this species, like in a couple of weeks. I don't know if you received that announcement. You probably know things better than this.
Maybe.
I'll send you that.
If I received it, I didn't check.
Yeah, I'll send you that.
So yeah, anyways, bioindicators, right? And then what are we indicating for? PAHs, okay? PAH stands for polycyclic aromatic hydrocarbon.
Oh.
There's a bunch of different kinds of PAHs, but the main thing that they have in common is they have these rings, right? This is called fluoranthene, and it has 4 rings, 1 pentagon and 3 hexagons. Those are called benzene rings. And individually by themselves, those are aromatic hydrocarbons. But then you put them together, and now it's polycyclic. You get it? So 2 or more is polycyclic, right? And they come from crude oil, right? So this is one of the main sources of PAHs in our environment. Okay, so fluoranthene is just one example. There's a bunch of other ones. Okay, so we're trying to use bioindicators to find out PAHs in the environment. All right, and that leads us to the paper that I'm going to discuss today by Vaezzadeh, right? Vaezzadeh. From a couple years ago, and they're trying to use oysters to monitor PAHs.
It's got to be from Australia.
No, it's not.
Really?
Malaysia?
Oh, Malaysia.
Yeah. Okay, so they're trying to use oysters, right? And why use oysters? Because this paper defines bioindicators, good bioindicators, by these categories right here. Okay, so the first one is it's abundant, easy to sample. Obviously, you don't want sampling difficulties to interfere with your research, right? Okay, the second one is it has to have a widespread distribution, preferably wider than the impact of the anthropogenic impact, right? So like, let's just say your toxic pollutant only goes so far, and if it goes further than the range of your species, then you don't know the whole range of the pollution, right? So if the range of the species is greater than the range of pollution, then you can actually figure out where the pollution is concentrated, right? So it has to have a wide distribution. You want it to be sedentary or sessile, because if it moves like a fish, then you have a contaminated fish that swims into a non-contaminated region, and then you catch that fish and find the contaminants, and now you think that area is contaminated, which is false, right? So if you have a sedentary organism, like an oyster, then, because they can't move, you know for sure that the contaminant is in the area. And if it's not contaminated, then there's no contaminant in the area, for sure, right? Because they would have gotten it. Okay, so it must be sedentary. It should be tolerant to the contaminants so that it doesn't die, and that if they all die, then you have nothing to study, right? And then the next one is you don't want it to break down the contaminant, right? Because if it breaks it down,
Touch screen.
yeah, I guess so. Yeah, if it,
Oh, cool.
I guess I don't need this. I can just touch the screen.
Awesome.
So you don't want to metabolize the contaminant because you want to see its uptake, right? So let's look at what Sandra is studying, right? The uptake of those nanoplastics inside the fish larvae, zebrafish. If they metabolize it, then you wouldn't even see it, right? So you don't want it to metabolize the contaminant. You want it to stay inside to check if it actually accumulated. And the last one is it should live long enough, because if it doesn't live long enough and dies right away, then you won't get to see the effect on different life stages, right? So here we have the mangrove. Now it doesn't work. Okay.
Hey.
So now we have the mangrove oyster, Crassostrea belcheri, right? Here's a picture. This is 2 centimeters, so it about fits in the palm of your hand. And this is a oyster that lives in the mangroves of Malaysia. And what they want to do is they want to take this and see if the pollution goes inside. So basically, the pollution that made its way into the sediments, the mangrove sediments, they want to see those pollutants inside the flesh of the oyster right over here. Okay, and so what they do is they got these study sites right here. See, this is Malaysia, and that's like Vietnam, Cambodia, and over here is Borneo and the Indo-Pacific. Malaysia is right here. It's a peninsula with these mangroves, right? Here they are: Merbok, Parai, Klang, Moor, and Puala. The most important one is Klang in the middle, because that one is the most urbanized, and hopefully that means more pollution and more PAHs in the area. We'll see if that's true or not, right? Okay, so the first step, right? The first step to this is get some sediment, find the PAHs in the sediment. Next step, get some oysters, find the PAHs in the oysters, and then find a correlation. In this case, they did Spearman's rho. All right, this is a basic correlation between those two things, and if they're correlated, then that means that the pollutants that are in the sediments made its way into the oysters, right? Pretty simple stuff. Okay, so how do you figure out how many PAHs are inside? Mass spec, right? Okay, so GCMS stands for gas chromatography mass spectrometry, and that's gives you this little graph here that tells you what compounds are inside your sample, right? And of course, safety first. You need to use something like internal standard, which is another compound that's similar to your PAH, that reacts in a similar way, and you need to make sure you know those things really well. Okay, you put them into the instrument, and you also put them into your processing, so that hopefully what happens during processing and what happens in the instrument happens to both your internal standard and your sample. That way, if you get any variations in your processing, or your instrument like has variations in each run, then you can detect those changes, and you don't think that those changes are due to true changes. In fact, they're just anomalies, right? So it's kind of like a calibrator. Does that make sense? Right? So you need to use these internal standards to check in case your machine malfunctions. It's not a true change, right?
Yeah, those are standards that the Sigma ostrich make those PAH standards. So we can basically buy them.
Yeah.
In there.
Right. Exactly. So those things, just put them in to check. Right? Okay, so now that they covered all their bases, right, we can figure out what PAHs are inside. We'll see what they found, right? Here's the sediment PAHs of the 5 different regions right here. Okay? And what do we see? mostly green, right? Green is 5 to 6 ring PAH and not that much blue, okay? 2 to 3 ring PAH. So if their hypothesis was correct, we should see this reflected in the oysters, right? Wrong. See, it's not reflected in the oysters. The oysters did not accumulate the high ones, the 5 to 6 rings, but they did accumulate the 2 to 3 ring ones. Okay, so what do we get? We see that the correlation was not very significant between the two, and the test kind of failed to show this, that it worked. They did get kind of desperate and say that, well, the high molecular weight PAHs, which is the 5 to 6 ring ones, showed correlation. Does it really? Doesn't really look like it. Okay, maybe the numbers worked out, but in my opinion, it doesn't really look like it. So at this point, I will need to warn you guys something that one of our very own professors said, right? You really have to consider the difference between statistical significance and practical significance. Okay,
He said that.
Right. See, in this case, the numbers worked out and it shows statistical significance, but I don't know if we can trust that, and it could just be a type 1 error. So, I don't know, practical significance, make sure we are on the same page with that one.
Well, you have to also consider like how much is in the water column at varying levels and how it's moving through the food chain.
Yeah, we should consider that, but the problem is they consider sediments instead. Right? See, water column would have been a good idea in this case.
Surface versus different depths, because the densities are different, right?
Yeah, and also considering how oysters feed, you probably want to use water data instead. Right, exactly. But I think possibly one of the reasons why they chose the sediments instead was the region is really has a lot of turbulence, and so the waters are very variable.
Is there a lot of crude oil mining in Malaysia that you know of?
No, I don't know of, but there could be.
I was reading an article that you might be interested in. It was talking about, it's called wastewater, but when they are getting the crude oil out, a lot of water comes out that is like has a lot of the pH attached to it.
Right.
And so in
during
these,
mining operations. For
yeah.
Sure.
And so it's like, then that it would be, I guess that's still water. I don't know if that would be like the sediment around.
Well, it'll go into the sediments, but as I'm going to explain later, they go into the sediments differently.
Okay.
How they go into the organism, right? So that would explain that better. Okay. So, anyways, let's do that. Right. Let's explain why they go into them differently. So they are concerned with different types of pHs, big ones and small ones, right? The big ones, we call them high molecular weight pH, and the low ones, the small ones, we call them low molecular weight pHs, right? So let's just compare the two real quick, the low ones and the high ones, right? This one over here is called phenanthrene, and this is Vaezzadeh, which is supposed to be the most well-known PAH. And so this one 5 rings, this one 3 rings, low, high, makes sense, right? Okay, so the main difference is the low molecular weight PAHs are characterized by being more soluble and more lipophilic, and the microbes decompose them easier than the high ones, and also in total it makes it more bioavailable to the oysters. Right, so there's a little bit of bias going on here. The high ones is the opposite. Less soluble, less lipophilic, the microbes don't decompose them, and they adhere to sediments. So can we explain this now?
Mm-hmm.
Yeah, because look, why are these so high in the sediments?
Because they're sticking to it.
Because they adhere to the sediments. Why are the low ones so low in the sediments?
Because they're broken down.
The microbes, the microbes broke them down, so you don't see that many. Okay, so how about over here? Why is there more of the low ones inside the oyster?
Are they metabolizing?
Oh, because it's more bioavailable. They're not metabolizing, or else you wouldn't see it. Right? Metabolism means breakdown.
Yeah, but pHs can be metabolized.
You mean they're breaking down the larger chain ones, maybe?
Maybe breaking down the large ones. That is, I guess it's possible, but the larger ones in general are harder to break down.
Well, usually for the larger ones, even if it's going to be degraded or modified, it won't directly turn into the smaller one.
It could be something different.
Yeah, it's a very different thing.
Yeah. And so, they're more bioavailable because they're soluble. They're in the water column, and also they're more lipophilic. And as you guys know, the toxins tend to stick to the lipids inside their body. So, I mean, if anything, the low ones that are more lipophilic are going to stick inside. Okay? So, as you guys can see, we can explain the problems with trying to use correlation between sediment and oyster tissue because of all those confounding factors. All right, so I would, in my opinion, correlation might not be such a good idea in this case, right? So overall, what I have to say is, did bioaccumulation happen? Now, of course it happened because you found the PAHs anyways, but not in the way you expected. You just got more of the low molecular weight ones. And then my recommendation, right? Maybe don't use correlation between sediments and oysters. Okay? So the first part talked about sediments versus oysters, and they normalized all of the sites by percentage, not by total amount. So this doesn't compare sites. This compares, this is just using those 5 sites of replicates, right? So how about let's compare the sites now, right? Compare the sites using this numbers data right here. Okay? We should be paying attention to Klang, the urbanized one. Is it different from the rest? Look how high those numbers are. It's very polluted in the sediments over in the Klang mangrove, right? Do we see this reflected in the oysters? Yes, a lot more than the oysters elsewhere, right? Now, one thing to point out real quick is this last one, lipid content. This was their attempt at controlling for oyster size, right? Because you don't want Klang to have the most because the oysters are the biggest, right? And it worked out because look, the oysters weren't not the biggest, and it still had the most. So lipid content probably factors into another reason why the correlation didn't quite work. So I say multiple regression would probably be a good idea in this case, right? Because there's so many different factors going in. That makes sense? All right. Okay. So we got all this. Right. Bioaccumulation happened. Klang definitely is more polluted, and it was found in oysters as well. So to some extent, the oysters did help, right? It was able to characterize a bit of the pollution in the mangroves. Okay, so that was part 1 of their study, right? Here's the main conclusions of the first part. The low and high molecular weight, they behave differently, so correlation might not be such a good idea. And the main reason for that is the oysters are filter feeders, right? They're probably going to get more of the low ones, which are more soluble, and not much of the high ones that are locked up in the sediment. If you do want to get the ones that are locked up in the sediment, because there were more of these than those, then you might consider a deposit feeder, like this sea cucumber right here. See, this is the conspicuous sea cucumber, and it's found in Hawaii, and I've lived there for several years, and I see these all the time. They live in mangroves. I don't know if they exist in Malaysia, but they live in mangroves, so if they might have, something to fulfill the same niche over there as well.
So you're saying maybe another species?
Yeah, maybe another. Oh, you mean in our lab?
Yeah, no, it's
well,
just
in this case,
right?
in this case, I would have to say that I don't really want to work with this. It's nasty. Yeah, these things are really nasty. So I would stick to the oysters.
I like the name.
Yeah, but yeah, I would not be surprised if Dr. Xu adds this. Okay, so
I did host a visiting scholar a while ago working on sea cucumbers, but this is just like japonica. There's a certain things
for aquaculture.
Around, yeah, for aquaculture.
To eat.
To eat.
Yeah. Okay.
Not this beautiful.
So. No. Trust me, this is not beautiful. Okay, when you see this in the wild, I describe these as like being bloodthirsty. These guys, look at them. They're red.
They're harmless.
This long.
That's kind of.
They're harmless.
Yeah. No, and you pick them up.
Yeah, they're
and
harmless.
They're like so weak. Like like a normal sea cucumber is firm, like an actual cucumber. But this one, just like all the water goes to one side. Like a really.
Like a slinky.
Yeah, kind of like a slinky. Yeah, exactly. So hang a slinky over your hand, and it droops all the way to the bottom. It's really nasty. And this part on the top turns like paper thin because there's nothing left there. Just,
No way.
yeah. Yeah, you just stretch it.
Interesting.
It's really nasty. It's super weak. Yeah.
Why
Okay, so moving on to part 2. Part 2 of their study is a really cool study about the origin and the source of the PAH. Okay. So the first one, they're trying to figure out the source, whether pyrogenic or petrogenic. I'll get into that in a second. And the second one is using BAF, the bioaccumulation factor, to another way to quantify bioaccumulation. Okay. Now, real quick, I want to let you guys know that BAF is calculated by the ratio between the amount of contaminants in the environment versus in the organism. So considering that they weren't very correlated, that part didn't really work very well. Okay, so we're just going to go ahead and skip that part. Okay, we already know the results, right? The sediment was not correlated with actual organisms. So instead we'll focus on that one, which is pretty interesting in my opinion. Okay, the source of the PAH found inside the oysters and the sediment, right? Okay, so how do you determine the source? There's two main sources, petro and pyrogenic. Petrogenic refers to anything involved with the crude oil itself. So the crude oil formation, mining activities, or it's a breakdown in the environment, okay, which causes PAHs to form. Petrogenic needs high heat. Okay, so this one's relatively low heat. High heat, such as internal combustion engines and incomplete combustion, leads to pyrogenic PAHs. Okay, and there's actually a third one, diagenic, which is natural processes. But of course, we're not interested. We're interested in the human processes, right? So here are these two main sources, petrogenic and pyrogenic, and how do we tell the difference, right? Well, the way that we tell the difference is we have to look at the types of PAHs, right? This paper tried its best to explain how it did it, and it was so confusing. I even had to ask Dr. Xu what this meant. Okay, let me read this to you guys. Based on the relative stability of parent PAHs, idena 1,2,3-CD pyrene and benzo-GHI pyrroline, which are mass 276 isomers, besides fluoranthene and pyrene, which are mass 202 isomers, have the widest range of stability and consequently can be properly used to distinguish petrogenic versus pyrogenic sources. Now here's the thing, I don't know what stability means, I don't know what a parent PAH is, and why do they use the word besides? Now this is just so confusing. And so let's take a moment to learn about PAHs, okay? So real quick, here's two different types of PAHs. This is called phenanthrene, and this is, what is that called?
Methyl.
That's methyl, right? So that is called methylphenanthrene. Okay, if you attach methyl to a phenanthrene, then this is called an alkylated PAH. And this one without the groups, that's the parent, okay? So this is the parent and this is the daughter. You can add methyl, ethyl, propyl, whatever groups to any of these edges and it will be an alkylated PAH. All right, and then the parent one is the one that doesn't have it. Okay, what's the difference? The parent one is more stable, all right? And when I say stable, I'm saying high temperatures. So if this is stable at high temperatures and this one's not, which one do you think is pyrogenic? It's going to be this one, because the high temperatures would have degraded these ones, and you wouldn't find them anymore, right? So this is the pyrogenic one, this is the petrogenic one, and if you see more of one or the other, hopefully you can figure out which one's which.
Cool.
Okay, so pyrogenic versus petrogenic. Okay, can we go try to figure out what this means now? Okay, so stability is referring to at high temperatures. Parent PAH is talking about the ones without the alkyl groups. Okay, and then because the alkylated ones and non-alkylated ones have different stabilities, then you can figure out petrogenic versus pyrogenic. That solves half of our problems. Why did they talk about isomers? See, that was so confusing. Isomers is a completely different process, right? I'll show you guys in a second. Right, going back to here, I just want to remind you guys real quick that the PAHs, methylated or not, or sorry, alkylated or not, are going to be found in all places, no matter what. They're always going to come in twos. You're not going to be able to say that, well, this place only has the parent ones, so that means it's pyrogenic. No, instead, you're going to always find both. It's the relative amounts that you need to be concerned with. So the relative amount, how you can discern different places, use something called a diagnostic ratio. And there's two types of diagnostic ratio. The first one being alkylated versus parent. Guess what the second one is? The isomer fraction. They're completely different, okay? But two different metrics to figure out whether it's pyrogenic or petrogenic. I don't know why they had to stick them in the same sentence. It confused me so much. It made me think that one of those was alkylated and the other one was not. But no, those are isomers. They have the same exact molecular weight. So they should have made it more clear. Or that paper was geared towards people who really understand PAHs.
Can the GCMS tell the difference between the isomers? Or no? Do you need something
Yes.
else? It can?
Yeah, I think so. Yeah, I'm guessing because they need to know how much of just fluoranthene
Yeah.
compared to both of them.
Yeah, they can
can
easily tell
distinguish?
you that. Yep.
Okay.
All right, there you go. Easily tell you that.
Yeah.
Yeah, that's
If we have that,
promising. Yeah.
if we have that equipment, we can easily find out.
The
But
chemistry department has one.
chemistry has
GCMS?
one, that's for sure.
They
Yeah.
just can't tell
Yeah,
the difference between a
that's
gnat
what it is.
tumors, right?
That's
Yeah.
what it is.
Like-
On the oysters, right? Okay, so anyways, alkylated versus parent and isomer fractions are both used to figure out petrogenic or pyrogenic sources. But here's the thing, this paper actually came out with a third diagnostic ratio, low to high molecular weight, which I think is totally wrong. You can't do this because there's so many confounding factors of why there might be more low or more high. And if they find any correlation between this and source, it's got to be coincidence. I don't even know why they mentioned that this was a way to tell. Right? This
I
is,
think another
this is
reason
wrong.
is like, they defined by themselves like what can be called a low molecular and what can be called a high molecular. There's really no clear cut, like this is low and this is high. So
Oh,
instead
I
they
see.
just they separate them by themselves. So that's why I feel like this method is it's pretty much what they try to convince people with the results they found.
Mhm.
So this is
Mm,
not
that's
something
fair.
like
Well, there is a difference
No.
between low and high though, because the 3-ring ones are definitely more found in organisms
Yes.
than the other ones.
yeah.
That's
mean,
true.
Are
Though-
those found more in the environment too, like in general? The three?
In the environment, it could be either or, depending on if you're looking at sediments or water. Remember? Yeah. Because in the sediments, you would see high.
Oh, yeah.
Yeah. So it kind of depends on what part of the environment you're
It
looking at.
varies. For this method, I figured it's going to be very a lot because even the
Yeah.
same products, if it's the same environment you found from the same environment, this ratio going to be changed. It's very different.
Right.
So it's not very stable. I don't
Yeah,
know.
I don't think so either. So I don't know why they mentioned that. Yeah. Okay. So let's just talk about the first two. All right. Alkylated versus parent and isomer fractions. Those two are tried and true and used in many different papers to tell pyrogenic or petrogenic sources. Okay. So how do you tell? These are the diagnostic ratios I was talking about. The first one is alkylated versus parent for MP over P. That's I forgot what that was called. Oh my god, I'm sorry. But methylphenanthrene,
Phenanthrene.
I think it's
Phenanthrene.
methylphenanthrene and regular phenanthrene, the parent, right? If it's greater than 2, it's petrogenic. If it's less than 0.5, it's pyrogenic. In between would be mixed sources, right? And this is from a different paper. As you guys can imagine, if you use a different alkylated and parent pH than phenanthrene, these numbers are going to differ as well, okay? And for isomer fractions, man, it took me so long to figure out why they wrote it like this. I was wondering why they gave unfair bias towards fluoranthene, but I was wrong. They just put it on the bottom because they need to get the total, right? So when you divide by the total, it's the fraction of fluoranthene for all 202 mass isomers. Okay, so less than 0.4, greater than 0.5, in between would be mixed, and obviously this is going to vary if you use different isomer fractions. For example, the other one, what was it called? Ideno 1,2,3-CD pyrene. That one, and its isomer is going to have different these numbers. Okay, so just make sure you know the correct numbers depending on which diagnostic ratio you want to use. Okay, you can use a whole bunch of different ones, right? And so, just to show you guys real quick, these are the two isomers, right? They're not structural isomers, they are mass isomers, right? So you can count the number of carbons, well, they're 16, same molecular weight as well. Okay, so anyways, using these ones, what did they find? Well, they came up with this fancy diagram. This is a cool looking diagram, and they're basically looking at the sediment and the oyster and seeing based off of the isomer ratio, and here it's unlabeled, but this is actually the alkylated versus parent ratio, to see where the source came from. Now, one quick criticism is it's really hard to tell what points these are without you have to constantly go back to the legend. And that's because they use different colors and different shapes. I would recommend that they use all the solid shapes for sediment and all the operation signs for oyster and have the same colors for the different sites. Okay, that would be way better. I think in this case they just had way too much fun in R, exploring the different possibilities, right? Because in here I really want to just see two matching colors being next to each other. That would show consistency. That would be good. So what do we see? Well, the thing that sticks out to me easiest is those two actually, dark and next to each other. Okay, the circle and the square, does it come from the same place? Yes, good, right? Okay, consistency. Now let's look at Klong, our famous polluted urbanized Klong, right? Right here, the minus sign and the purple X. Very different results here. This one means that the alkylated versus parent pH ratio shows that the oyster comes from a mixed source and that the sediment comes from petrogenic. Okay, but if you look at the isomer ratio, it shows that the sediment is actually combustion mismatch, and also it shows that the oyster is from combustion as well. Okay, so we see some disagreement here. Okay, and initially you might think that this disagreement is bad because if you don't get converging results, then they don't agree and therefore they're both wrong, right? But no, actually I think they're both right. Okay, I think that by using two different sample types, sediment and oyster, and also two different metrics, isomer ratio and the alkylated versus parent, we're able to get the full picture because maybe one of them can only detect a certain thing and the other one detects the other thing that was missed by the first one. So I actually think it's a good idea that they did to use both sediment and oyster and also to use both of those metrics to determine diagnostic ratios to determine the source. Right? So I could see some logic in why they chose oyster, which is a filter feeder, and sediment because they cover two different bases. But what they shouldn't have done is try to correlate those two. Does that sound okay? All right, so that's it, right? They found the sources. Hopefully the only way to tell is if you did some sort of isotope tracing, which we didn't do, right? They just inferred these locations, these sources. Okay, so overall, the two main points is there was evidence for oyster bioaccumulation. Good, right? Oysters could in part be used to show be a bioindicator, right? Second is tracing the source of the PAHs kind of worked, right? I mean, you just had to test to make sure it was true, but I'd say that, doing multiple sample types and multiple metrics was a good idea to do this, right? But I can't end without telling you about a couple things that I think they need to fix, right? So again, reiterate, don't use correlation between sediments and oysters because this one is a filter feeder, right? The BAF is not going to work for the same reasons and also all those confounding factors. Use a deposit feeder for sediments if you want to correlate, and label your graphs better with the proper recommendations, and also the low to high molecular weight ratios is not going to work at all. Okay, those ones is too many confounding factors, and they're not going to indicate the source. They're just coincidentally correlated. Okay, so that's it. Hopefully that makes sense. But if you guys have any questions, I feel like I'm an expert at PAHs now.
Does he really know more than you know?
Well, keep it low, okay? Yeah, you did present like really well for the basic knowledge for PAHs. I kind of realized this is also something that we can think of.
Yeah.
I know for your project you're basically focused on the petrogenic resource for the PAHs.
Yeah.
But it might be also interesting if we're also trying to identify if those accumulations inside the oyster could be possibly from the pyrogenic.
Yeah. Well,
other solution.
whether it's the source, we can find out the source or not. The reason why I chose this paper was I was hoping to propose to Dr. Xu that the second part of the research that I was talking about. Remember, I was talking about we should go collect samples, water samples, and oysters from other farms, and find the PAHs.
So I had another idea. I want to really talk to you about more PAHs. I've been trying to do a lot of research about it because I also find them really interesting. I've been looking at it from a little bit of a different view. I was reading a paper and it was talking about the point sources.
Oh,
and so you could almost you could go to point source,
Exact.
like, so you could do like your in situ experiments, and then you could go and collect,
Yeah.
like in the actual environment,
Mhm.
and see as well.
Oh, okay.
Interesting to see like water samples from the Harbor Bridge area.
Oh, yeah.
So like I was trying to relate it to like my marine snails, where we could go
Oysters there.
and take from like the Corpus City refineries, we could do along the whole coast, Texas City, Houston, and we could go to those point sources and actually see how that is affecting them in the environment. There's also some really cool stuff that they're finding, high rates of PAHs in our sewer water now.
Awesome.
Sounds like
So,
you guys
and
can
we have sewer water, so, but we don't have the identified PAHs. But we should bounce ideas off each other,
Yeah.
because this is some really cool research that I think we could create.
sounds like they've been sitting in the lab for too long. They all want to go out for
Yes.
some fun.
Yeah.
Yeah, we'll find a
It'd be cool to like, do some like GIS mapping too.
Oh, I see.
I don't
Yeah.
know, like we could do like the Biomonitors.
just last summer Simon guys has
Yeah,
multiple
really good.
opportunities to go out.
Yeah.